Friends, welcome to Biology Made Easy. I hope you are all well. So today in this video we'll discuss the dihybrid cross. After being successful in monohybrid cross, Mendel carried out experiments taking two characters at a time. So when a cross is made between two plants which differ in two contrasting characters, that is known as dihybrid cross. In one of the dihybrid crosses, Mendel took the shape of the seed and the color of the seed. So as you know, the shape of the seed may be round or wrinkled, and the color of the seed may be yellow or green. So he made a cross between a plant with a round and yellow seeds with another that is wrinkled and green seeds. A cross was made. So this is capital R, capital R, capital Y, capital Y. This is small r, small r, small y, small y. Because this is homozygous for round and yellow, and this is the wrinkled green also in homozygous condition. Now when the cross is made in the F1 hybrid, it gave rise to a plant having the genotype. So what will be the nature of this plant? The nature of this plant will be round and yellow seeds. Now when this F1 hybrid was intercrossed, the F2 generation was obtained. So what is the F2 generation? Let us draw the square, the Punnett square, so that we can know the details. When the Punnett square the four types of gametes which will form the male side and this side the female side. So this will be capital R, capital Y, capital R, small r, capital Y, small r, small y. Similarly in this side it will be capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. Now the possibility is that the 16 types of plants will be produced here. So you see here it is capital R, capital Y. That means this is when capital R is there, it will be round, the seed will be round, and when capital Y is present, it will be yellow in color. That means all these will be round with yellow in color. Similarly, here capital R, capital Y is there. That means all in this horizontal row, they will be round with the yellow seeds. Then you see another place, this one. From this place it will come from the female side capital R, from the male side capital Y. So this will be round with yellow seeds. And another place you see this one. From male side capital R, from a female side capital Y. So this will be also round with yellow seeds. Now you see how many of these are round with yellow seeds. 1, 2, 3, 4, 5, 6, 7, 8, 9. That means the 9 by 16 are round with yellow seeds. Then you see what is happening at this place. At this place it will be capital R, small y, small y. Similarly, at this place it will be capital R, small y, small y. And at this place it will be capital R, small y, small y. So, when it is capital R, it is round. But when y is in the lower case, it will be green in color. That means this is a round and green. This is round and green in color. Then you come to these places. At these places what will happen? We will find here it is smaller, capital Y. Here smaller, capital Y, small y. And here it will be smaller, capital Y, small y. Now see, here small r is present, in lowercase r is present. That means it will be wrinkled. But capital Y is present. That means it will be yellow. That means in these cases, these sheets will be wrinkled and yellow. And what about the last one? The last one will be smaller, small y. That means both the recessive characters are present. That means this will be smaller, means it will be wrinkled. And small y, that means it will be green in color. That means this is wrinkled and green in color. That means, now see, 9 by 16 round and yellow, and 3 are round and green in color, 3 by 16, then 3 by 16 are wrinkled and yellow, and 1 by 16 is wrinkled and green in color. That means there are 4 types of phenotypes present in the ratio of 9:3:3:1. And in monohybrid class we have discussed that the phenotypic ratio is equal to 3:1 to the power n, where n is equal to number of contrasting characters. So here it is 3:1 and number of contrasting characters are 2. Now you see this is coming in the ratio of 9:3:3:1. That means 4 types are there. So this is the phenotypic ratio. Now from this we can get the genotypic ratio. So what is the genotypic ratio? Now you see at this place it will be capital R, capital Y, capital Y. Here it is capital R, small y, smaller, capital Y, capital R, and this. That means at these 4 places, these 4 corner to corner, these are homozygous plants. That means these 4 are homozygous plants, while all others are heterozygous. You see, capital R, capital Y, capital R, capital, small y. Here smaller, capital Y, smaller, small y. This, out of these 16 plants, 4 are completely homozygous and the remaining 12, they are heterozygous. Now, if this is the phenotype. Now, if you get the genotype. Now, again we know that the genotypic ratio is 1:2:1 to the power n. So here n is equal to 2. So 1:2:1, 2. That means it will give rise to ratio that is 1:2:1, 2:4:2:1:2:1. That means it is giving rise to 9 types. That means the phenotypes are of 4 types, but the genotypes are of 9 types. Now, what are these 9 types of genotypes? Let us see. Now, this it will draw like this, so we can better understand the things. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now you see. Now here this capital R, capital Y, capital Y. Capital R, capital Y, capital Y. It is only 1. Then capital R, capital Y, and 1 small y. This will be, the number will be 2. Then capital R, small y, the number will be 1. then capital R, small r, capital Y, the number will be 2. Then capital R, small r, and capital Y, small y, it will be 4. Then capital R, small y, this will be 2. Then small r, capital Y, 1. Then small r, capital Y, 2. Then small r, small y, 1. That means the genotypic ratio, which is 1:2:1:2:4:2:1 to the 9 types. These are the 9 types of the genotypes. Now from this again, we can derive certain conclusions. What are the conclusions? How many are pure homozygotes? How many of these are pure homozygotes? So say pure homozygotes. Pure homozygotes are only 2. And what are these 2? One is capital R, capital Y, this number 1, and the last one, that is small r, small y. This is dominant and this is recessive. That means pure homozygous. But how many homozygous plants are there? How many homozygous plants are there? 4. You can see this corner to corner. That is, what are these 4 types? Capital R, capital Y, the second one. The second one is capital R, small y, the third one. The small r, capital Y, and the last one. The small r, small y. So, these are the pure homozygous plants. Now, how many of these plants are coming in the, what is the ratio of homozygous to heterozygotes? Homozygous is to heterozygous. So from these 16 plants, 4 are homozygotes, while 12 are heterozygotes. That means the homozygous, heterozygous, they are coming in the ratio of 1:3. You can see it. These 4 are homozygous, the others are heterozygotes. And how many of them are pure heterozygotes? Pure, or we can call double heterozygotes. That will be still better. How many are double heterozygotes? Double heterozygotes are only 4 in number. And what are these 4? These are capital R, small r, capital Y, small y. Double heterozygotes. So in this way, several questions can be asked on this also. So this is how we are getting the phenotypic ratio and genotypic ratio. Now from the findings of this, we can see certain things. So what are the findings of the result we can see? So let me erase this one. So if we will find the results or findings of the dihybrid cross, then what we are getting? What we are getting means what Mendel got. That means it is found in F2 generation, the phenotypic ratio is 9:3:3:1, out of which 9 both dominant. You can see the 9, this 9, the round and yellow ones, both dominant. At 3 places, 1 dominant and 1 recessive. like this. One dominant, one recessive. Here also one dominant, one recessive. The other three, one recessive and one dominant. And at one place, both recessive. So this is the findings of this Mendel's dihybrid. This is one. Number two, in the F2 generation, the plants which we obtained, out of these, two are parentals. Two are parental types. So what are the parental types here? Now here we got the round and yellow. This is one parent, and the other is wrinkled and green. Two are parental types, while two are recombinants. Two are recombinants. So what are these recombinants? That will say the round combined with the green. Round with the green, and the other is that is a wrinkled with yellow. Wrinkled and yellow. So this is the second findings. Now come to the third finding. What is third finding? How it is possible Mendel took two contrasting characters. One is the shape of the seed, the other is the color of the seed. But how one of the that is the shape of the seed combined with the color and vice versa. How is it possible? That means the inheritance of the shape of the seed and the color of seed are independent of each other. This is the vital point. The next point is inheritance of seed shape. Inheritance of seed shape and seed color are independent of each other. That is why the round color also, the round shape also combined with the green one, and the wrinkled one also combined with the yellow color. It is because of the independent assortment and random fertilization as shown in the Punnett square. From this finding, the law of independent assortment was derived. So these are the important findings. Now after this, we have to see how Mendel applied mathematics to his experiments, mathematical analysis, because that is very important for Mendel, for which he was not recognized. So Mendelian analysis in mathematics. Now number one, the possible different types of gametes, possible different types of gametes is 2 to the power n, where n is equal to number of contrasting characters. So here number of contrasting characters is 2, so the possible different types of gametes is 2 to the power 2, 4, and we have seen the 4 types of gametes. The second mathematical analysis, that is possible different types of phenotypes. It is again 2 to the power n. So here is the n is now the contrasting character. So again it is 4 possible phenotypes, 4. So we have got the 4 phenotypes, 9:3:3:1, 4 types of phenotypes we have got. Then the third one, the possible different types of genotypes, it is 3 to the power n. Now again you see here n is number of contrasting characters. So n is contrasting characters means it is 2. That means it will be 3 to the power 3. That means it is 9. And now we have seen that there are the genotypes are 9 types. 1:2:1, 2:4:2, 1:2:1. Already we have written that one. Now the next mathematical analysis, the phenotypic ratio is 3:1 to the power n, where n is called number of contrasting characters. So here again n is equal to 2. That means here it is seen that 3:1 square. That means again the phenotypic ratio was 9:3:3:1. So this is the formula. Then the next mathematical is the genotypic ratio. So it is 1:2:1 to the power n. So here n is equal to 2. That means 1:2:1 to the power 2. That means when this will be its square will give rise to 1:2:1. Then 2:4:2. Then 1:2:1. So these are the 9 types of genotypes. So this is the mathematical formula he applied to his dihybrid cross. So this is about the dihybrid cross. Now let us discuss very briefly about a trihybrid cross.